everyone. Welcome back to the boot camp. Today I'm going to talk to you about unsupervised learning techniques. My name is Zhiping Weng, and I'm professor and director of the program in bioinformatics and integrated biology. The slides for today's lecture have been kindly made by my colleague, Dr. Joe Moore, who collaborates with me on many large-scale genomics projects. After you have collected some experimental data, you might want to apply statistical learning techniques to analyze your data. What kind of techniques you can use depends on what kind of data you have. One type of data has a response or an output. For example, RNA-seq data on tissue samples that are labeled as disease or control. You may perform supervised learning on such data. Other types of data are not associated with a response or output. For example, in your RNA-seq data from tissue samples of patients. However, you do not know what kind of disease the patients suffer from, so you cannot label your data. For such data, you can only perform unsupervised learning techniques, which are the focus of today's lecture. Today, we are going to cover two types of widely used unsupervised learning techniques: clustering and dimensionality reduction. So here is the outline for today's lecture. In part 1, we will cover two widely used clustering techniques: k-means clustering and hierarchical clustering. In part 2, we will cover two techniques for dimensionality reduction: principal component analysis, often seen with the acronym PCA, and another technique called TISNI. When you perform clustering, you actually assign data points to groups based on how similar they are to each other. Why would you want to cluster your data? You might want to explore the underlying structure in your data. It is also very useful for quality control purposes. You might have heard of batch effects, which are artificial reasons that make your data clumped into groups. For example, RNA-seq experiments performed on the same day may appear to be more similar to each other than the experiments performed on different days. And clearly, the difference is not biological. The first clustering technique we will talk about is k-means clustering, where k indicates how many clusters you wish to have. We're going to explain this using a simple one-dimensional example, whereby we have 12 data points on a line. For this example, we're going to set k to be 3. We randomly pick 3 data points as the centers for our 3 clusters. For each data point, we then compute its distance to each of the 3 cluster centers, as indicated by the length of these 3 colored lines. And then we pick the shortest distance. In this case, for the first data point, the shortest distance is the blue line. So we pick that cluster, corresponding cluster, to assign our first data point to. We assign the first data point to the blue cluster. We repeat this process for the third data point, and we can see that it is closest to the green cluster center, so we assign it to the green cluster. So we repeat for all other data points and assign each to its closest cluster center. Once we have completed the assignments for all the data points, we need to recompute the position of each cluster center based on the assignments. So for the blue cluster, we take the 2 data points belong to it and average their position. And then we get this blue line that indicates the new cluster center. The new cluster center is the average or mean position of the data points that belong to it. Therefore, the technique is called k-means clustering. So we have these blue, green, and yellow vertical lines to indicate the cluster center of the 3 newly defined clusters. Now we can start again to perform the next round of assignments. At this time, we're going to use the mean positions of the 3 clusters centers. So we pause for a moment to check. Did any of the cluster assignments change for the data points? In this example, the answer is no. All the data points still belong to the same cluster as in the previous round of assignment. If the answer would have been yes, then we would need to repeat steps 6 and 7 until the cluster assignments no longer change. So with all points assigned to the 3 clusters, we have successfully performed 1 round of the k-means clustering. Now we can evaluate how well we did. So the performance is evaluated by calculating the variation among the points within each cluster, as illustrated by the 3 colored horizontal bars. You can see that the brown cluster has the highest variation, indicated by the longest brown bar. We can plot the 3 horizontal bars together to each other, next to each other, to indicate the total variations of this round of k-means clustering. We then repeat the entire process using randomly selected 3 data points again as the starting cluster centers. We assign each data point to the closest cluster. We update the cluster centers based on the assignment, indicated by the 3 vertical bars. We assign all data points to the updated cluster center. We update the cluster center again. Since there were changes in the assignment, we have to perform 1 more round of assignments of the data points to their nearest cluster center. Now we can see there are 3 tightly formed clusters. Since the cluster assignment no longer changes, there is no need to iterate again. The second round of clustering performed better, illustrated by the smaller total variation among the data points that belong to the same cluster, compared with the first round of clustering. So you can see round 2 has a shorter length for the total line compared with round 1. For a real application, we typically perform many rounds of clustering, in the order of tens of thousands of rounds. Finally, we look through all the runs and choose the one with the lowest total variation, which is round 2 in this case. Well, you may ask, how do you decide what is the most appropriate value for k? The solution is actually to try different values of k. In this example, we're trying k equals 1, k equals 2, k equals 3, k equals 4. For each k, we can plot the total variation on this two-dimensional graph. Naturally, when you have a larger k, you're going to have a smaller total variation. Because if you think about it in the most extreme case, if you assign each data point to its own cluster, then the total variation will be 0. Having that in mind, we're looking for an inflection point in the graph, also known as the elbow, so that you can choose the best k. For this example, the most appropriate k value is 3, since when you further increase k, you did not bring down the total variation much. Our one-dimensional example can be easily extended to higher dimensions. Here is an illustration in 2 dimensions. Again, in our example, k equals 3. In step 1, we randomly generate the initial positions of the 3 cluster centers, as indicated by the red, green, and blue circles. Note, these are not actual data points in this example. They don't have to be, they can be. In step 2, k clusters were created by associating each data point to the nearest cluster center. In this illustration, the space is partitioned according to the Voronoi diagram generated using the 3 cluster centers. In mathematics, a Voronoi diagram is a way of partitioning the space into regions that are close to a given set of points. In our case, the 3 cluster centers. In step 3, the new assignments of all data points are then used to update the cluster centers. We repeat steps 2 and 3 until convergence. Here is a biological example of k-means clustering at work. In this 2015 paper, data were generated on mRNA translation and protein levels in HeLa cells across the cell cycle. K-means clustering was used to discover 5 groups of genes with different patterns in these 3 types of data across the cell cycle. So that ends our first section of the course. And now we're going to switch gear to talk about the second type of clustering technique, hierarchical clustering. We're using a biological example. Suppose you have treated some cells with a chemical, and then you performed RNA-seq at 3 time points, 0, 30, and 60 minutes after the treatment. You want to know whether some genes show similar expression patterns in your time course experiment. The first step of hierarchical clustering is to find out which pair of genes has the most similar expression patterns. We have to examine all pairs of genes. We can see that gene 1 and gene 2 are dissimilar. Gene 1 and gene 3 are very similar. Gene 1 and gene 4 are dissimilar. Gene 2 and gene 3 are dissimilar. And finally, gene 2 and gene 4 are similar, although at a lesser degree than gene 1 and gene 3. Finally, gene 3 and gene 4 are dissimilar. So we group the two genes with the most similar expression patterns together to form cluster 1. Note, we have swapped the rows for gene 2 and gene 3 so that we can place gene 3 right below gene 1 to form cluster 1. Now we repeat the process while treating cluster 1 as a single entity. Again, we examine the similarity between all pairs of genes / clusters. Cluster 1 and gene 2 are dissimilar. Class 1 and gene 4 are dissimilar. Finally, gene 2 and gene 4 are similar. So we group gene 2 and gene 4 into cluster 2. We repeat this step while treating cluster 1 and cluster 2 as individual entities. There is only one pair left, so it has to be the most similar pair. Accordingly, we group cluster 1 and cluster 2 to form cluster 3. At this point, we have completed our hierarchical clustering exercise. The result of a hierarchical clustering can be displayed using a dendrogram, where the structure of the dendrogram indicates the order of cluster formation, while shorter branch lengths indicating higher similarities between the two corresponding entities, be it genes or clusters. How do we calculate similarity between two entities? Or on the other side of the coin, how do we calculate the distance between them? Here are two commonly used distance metrics: the Euclidean distance, or the straight line distance as the crow flies, and the Manhattan distance, or the city block distance as the cab drives. How do we apply a distance metric to compare a cluster with a gene? There are 3 ways. First, take the center position of each cluster and compute its distance to the gene. This method is often called the centroid, or average linkage. In the second approach, we can take the data point in the cluster that's closest to the gene and use it to compute the distance to the gene. This second method is called single linkage. Generally speaking, single linkage is not recommended because it tends to generate artificial-looking clusters. In the third approach, you can take the data point in the cluster that is furthest away from the gene and then use it to calculate the distance. This method is called complete linkage. If you use the hclust function in the R program to perform hierarchical clustering, by default, it uses complete linkage, unless you specify one of the 2 other choices. Here is a biological application of hierarchical clustering to analyze differential gene expression between 2 colon sections, the crypt section and the top section, indicated by blue and pink colors, respectively. You can color genes as well as samples. As you can see, experiments from the same colon section cluster tightly together, showing distinct gene expression patterns between the 2 sections. When we zoom in to 2 clusters of the genes, we can see example genes known to be important for colon function. Here is another example for applying hierarchical clustering to phylogeny, the study of evolutionary relationships among species. Flaviviridae viruses infect humans and animals. Hierarchical clustering of different species of Flaviviridae viruses by their codon usage of protein coding genes reveals that species tend to cluster by the types of hosts they infect. So that concludes our discussion on hierarchical clustering. Now we switch gear to talk about the second type of unsupervised learning technique, dimensionality reduction. Many biological data sets have a large number of features, which are dimensions in the parlance of statistical learning. For example, an RNA-seq dataset has tens of thousands of genes for which you have measured the expression levels. Another example is that you have sequenced the genomes of multiple individuals, and you want to use the whole genome sequence for predicting the propensity that an individual will contract a disease later in life. And the billions of genomic bases will each be treated as a dimension. Working with high-dimensional data has many challenges. For example, some of these dimensions may be noise, and the distance between data points in high dimensions have undesirable properties, which might break many statistical techniques that work well in a lower dimension. Dimensionality reduction allows us to project high-dimensional data to a low dimension. In particular, projecting data onto 2 dimensions can also greatly facilitate the visualization and the biological interpretation of your data. Principal component analysis is a technique that uses a smaller number of representative dimensions, each of which is a linear combination of the original dimensions of the data, to explain most of the variance that exists in the data. In contrast to the next technique we will discuss, t-SNE, PCA focuses on keeping distant data points, as measured in a high-dimensional space, far apart in the low-dimensional space as well. Here is a 2-dimensional example to illustrate how PCA works. There are 6 cell types for which we have measured the expressions of 2 genes, hence 6 data points on 2 dimensions. We first plot the data as 6 colored circles. In the next step, we find the average expression levels across the 6 data points, as indicated by these 2 gray crosses on the 2 axes. Then we use these 2 averages as the x and the y coordinates of the centroid of our data, indicated by this black cross. The centroid of the data will become our new origin. Effectively, we move our data points so that their centroid is at the origin of our coordinate system. The next task of PCA is to identify a line that passes through the origin and minimizes the distance between all the points and the line. The distances are indicated by these black lines with arrows. The best line will be the first principal component, abbreviated as PC1, shown here as the maroon-colored dashed line. As you can see, all the data points are very close to this line, indicated by the short black lines with arrows. By design, principal component 2 must be orthogonal to principal component 1 and also must pass through the origin. In our simple two-dimensional example, there is only one line that can possibly fulfill these two requirements, as indicated by this blue dashed line. Once we have identified the two principal components, we're going to map each data point onto these two new axes. The resulting new coordinates are indicated by the colored crosses on the two principal components. Now we are going to rotate our coordinate system such that principal component 1 becomes the new x-axis and the principal component 2 becomes the new y-axis. Once we have a new coordinate system, now we can put back our 6 data points one by one according to their coordinates in the new system. After that it's done, congratulations, you have now successfully performed PCA and plotted your data on the two principal components. As you may recall, the goal of PCA is to explain most of the variations in your data. So now we can ask, how much variation in your data does each principal component explain? To answer this question for PC1, we first calculate the sum of the squared distance between the projection of each data point and the origin for PC1. Then we divide this sum by the number of data points less than by 1. We can do the same thing for PC2. We then sum together the variations for all the PCs and calculate their respective fractions. In our two-dimensional example, PC1 explains 93.6% of the variation in your data, while PC2 explains the remaining 6.4%, as shown in this bar plot on the right. The bar plot is also called a ski plot. Now we can expand our scheme to higher dimensions. For example, now for 3 genes. Again, we plot the data. We find the means for gene 1, gene 2, and gene 3. And these 3 means will specify the center of the new coordinate system. We perform the 3 steps as in our previous two-dimensional example to find the best line that minimizes the points, minimizes the distance from the points to the line, which will give us PC1. Then we find the best line that's orthogonal to PC1, with the best being the minimal total distance between the data points and this new PC2 line. So that will give us the PC2. Unlike in the two-dimensional example, now we actually have a choice for PC2 because there is a whole plane that is perpendicular or orthogonal to PC1, and you need to find the best line in this plane so that you can get PC2. And finally, PC3 is specified for us because it needs to be orthogonal to both PC1 and PC2, and we started out with three-dimensional data. Again, we project each data point to the three PCs and plot them in the new coordinate system. We can calculate the variation explained by each PC as in our two-dimensional example, resulting in this ski plot with 3 bars. You can see from the plot, PC1 and PC2 together explains 98% of the variation in our data, suggesting that we can toss away PC3 without sacrificing much. By doing so, we can now visualize our data in the 2 dimensions specified by PC1 and PC2, and we have succeeded in dimensional reduction from 3 dimensions to 2 dimensions. Two dimension is a plane, and it's very clear to visualize data points on 2 dimensions. We can even expand to higher dimensions. For a typical PCA analysis of RNA-seq data, we can have up to 50,000 genes, which would include both protein coding and non-coding genes. That would give us 50,000 dimensions. But don't you worry, because your computer will happily perform PCA in the same way, except it's going to take a bit longer. Here is a biological example of clustering tissue samples based on their gene expression, with the data taken from the GTEx consortium. Each dot in the scatter plot indicates one tissue sample, and its color indicates its tissue of origin. There are many dots with the same color. These are the same tissue taken from different donors. The two-dimensional scatter plot with PC1 and PC2 as the 2 axes indicates that samples of the same tissue from different donors tend to cluster together, which is consistent with their having similar gene expression profiles. That concludes our discussion on PCA. Finally, we are going to cover the last topic of today's lecture, a technique called T-distributed stochastic neighboring embedding, TSNE in short. Well, you may wonder how to pronounce this acronym, and the majority opinion is to pronounce it like a word, TSNE. The math behind this technique is a bit involved. If you want to get more information, you can watch this Google Tech talk, which does a very good job explaining the gist of the method. If you want even more information, you can read the original paper published in the Journal of Machine Learning Research in 2008. You might be interested to know that this paper has been cited for more than 15,000 times. The main idea of TSNE is to project high-dimensional data onto 2 dimensions while preserving the local structure. Note that I mentioned earlier that a goal of PCA is to keep distant data points far apart in the lower dimension, but PCA cares less about preserving the local structure of points that are near each other. To illustrate the point, we are going to use a simple example of reducing 2-dimensional data onto 1 dimension. As you can see here, if we choose either the x-axis or the y-axis as our 1 dimension, the total structure of our data points illustrated by the 3 clusters in red, yellow, and blue. It's kind of messed up. So let's not do that. So let's start with a brand new one dimension, which looks like a number line, hoping that we can place the data points onto this one-dimensional number line while preserving the 3 clusters. To do this, we first place our data points randomly on the number line. Then we move the data points on the one-dimensional number line one by one to a new position so that they can maintain the similarity to the other data points as specified by the original 2-dimensional space. We will have to do that in many steps until the algorithm converges. Well, how do we determine the similarity between data points in a one-dimensional number line? For each point, we're going to calculate its distance from all other data points using the original 2-dimensional space. These distances are then plotted against a normal distribution. The normal distribution is the bell curve that is commonly used to discuss things like heights, weights, or body mass index of individuals in a population. Using this normal distribution, we can obtain a normalized score for the similarity. Data points that are close to each other, for example, if we use the dark blue point to calculate the distances, the light blue points are close to the dark blue point. These light blue points will get a higher similarity score than other points that are far away, such as the red and the yellow points. Performing this calculation for each data point will allow you to create a similarity matrix between all pairs of points, as shown in this matrix that looks like a heat map on the right. So that is using the original 2 dimensions. We can do exactly the same and create another similarity matrix between all the points that we have placed on the one-dimensional number line. However, now we don't use the normal distribution. Instead, we're going to use the t-distribution. Well, t-distribution may look like a normal distribution. It's not as tall in the middle, and it has fatter tails. The lower middle in the t-distribution will prevent overcrowding of data points near the origin of the coordinate system, which will allow us to model the local structure of the data. So the t-distribution indicates the name in the t-SNE. Now we have 2 similarity matrices. The one on the left, which is generated from the points on the one-dimensional number line. The matrix on the right, which was generated using the points in the original 2-dimensional space. Using individual moves of the points on the one-dimensional number line, the t-SNE algorithm makes the left matrix look more and more like the right one. I must stress that I'm giving you a very simplified explanation of the t-SNE algorithm. If you want to use the algorithm, you need to understand it more deeply. Make sure to watch the Google Tech Talk. Most importantly, you need to know concepts such as perplexity and normalization, and can choose these parameters appropriately for your biological application. Also, as t-SNE, the name implies. It is a stochastic algorithm because of the random assignment of the data points to the number line. Therefore, you can get slightly different results every time you run t-SNE. Here is a biological example that plots single cells on the two t-SNE dimensions according to single-cell RNA-seq data. 5 clusters are apparent, shown in different colors in panel A. The remaining panels indicate the expression levels of marker genes, which are known to be expressed at higher levels in certain tissue types, but not in other tissue types. We have discussed 2 techniques for dimensional reduction, PCA and t-SNE. How do they compare with each other? As I mentioned above, PCA is better at placing data points that are far apart, while t-SNE is better at placing data points that are nearby. PCA is easier to interpret than t-SNE, while t-SNE works better for data with nonlinear relationships. Actually, PCA is a very general technique and is often used as a pre-processing step to other techniques. For example, you might be interested in knowing that we often perform PCA first. Take the first 30 or so principal components, and then we run t-SNE on top of that. If you are interested in the things that we have been discussing today, Joe and I teach this course BBS 741. If you click the title, you will get to the course syllabus. We teach it every fall semester. We're actually teaching it right now. Here is a list of the topics. You can see that the 3rd lecture is devoted to PCA, and the 4th lecture covers t-SNE and another widely used technique called UMAP. Well, that's all I have to cover today. Thank you very much for listening.